Section 10.1, Cloning and Genetic Engineering. By the end of this section, you will be able to explain the basic techniques used to manipulate genetic material and explain molecular and reproductive cloning. Biotechnology is the use of artificial methods to modify the genetic material of living organisms or cells to produce novel compounds or to perform new functions. Biotechnology has been used for improving livestock and crops since the beginning of agriculture through selective breeding. Since the discovery of the structure of DNA in 1953, and particularly since the development of tools and methods to manipulate DNA in the 1970s, biotechnology has become synonymous with the manipulation of organisms' DNA at the molecular level. The primary applications of this technology are in medicine, for the production of vaccines and antibiotics, and in agriculture, for the genetic modification of crops. Biotechnology also has many industrial applications, such as fermentation, the treatment of oil spills, and the production of biofuels, as well as many household applications, such as the use of enzymes in laundry detergent. Manipulating genetic material. To accomplish the applications described above, biotechnologists must be able to extract, manipulate, and analyze nucleic acids. Review of nucleic acid structure. To understand the basic techniques used to work with nucleic acids, remember that nucleic acids are macromolecules made of nucleotides, a sugar, a phosphate, and a nitrogenous base. The phosphate groups on these molecules each have a net negative charge. An entire set of DNA molecules in the nucleus of eukaryotic organisms is called the genome. DNA has two complementary strands linked by hydrogen bonds between the paired bases. Unlike DNA in eukaryotic cells, RNA molecules leave the nucleus. Messenger RNA, or mRNA, is analyzed most frequently because it represents the protein coding genes that are being expressed in the cell. Isolation of nucleic acids. To study or manipulate nucleic acids, the DNA must first be extracted from cells. Various techniques are used to extract different types of DNA. Most nucleic acid extraction techniques involve steps to break open the cell, and then the use of enzymatic reactions to destroy all undesired macromolecules. Cells are broken open using a detergent solution containing buffering compounds. To prevent degradation and contamination, macromolecules such as proteins and RNA are inactivated using enzymes. The DNA is then brought out of solution using alcohol. The resulting DNA, because it is made up of long polymers, forms a gelatinous mass. Figure 10.2. This diagram shows the basic method used for extraction of DNA. First, cells are lysed using a detergent that disrupts the plasma membrane. Then, cell contents are treated with protease to destroy protein and RNase to destroy RNA. Cell debris is pelleted in a centrifuge. The supernatant, or liquid, containing the DNA is transferred to a clean tube. Finally, the DNA is precipitated with ethanol. It forms viscous strands that can be spooled on a glass rod. RNA is studied to understand gene expression patterns in cells. RNA is naturally very unstable because enzymes that break down RNA are commonly present in nature. Some are even secreted by our own skin and are very difficult to inactivate. Similar to DNA extraction, RNA extraction involves the use of various buffers and enzymes to inactivate other macromolecules and preserve only the RNA. Gel electrophoresis. Because nucleic acids are negatively charged ions at neutral or alkaline pH in an aqueous environment, they can be moved by an electric field. Gel electrophoresis is a technique used to separate charged molecules on the basis of size and charge. The nucleic acids can be separated as whole chromosomes or as fragments. The nucleic acids are loaded into a slot at one end of a gel matrix. An electric current is applied, and negatively charged molecules are pulled toward the opposite end of the gel, the end with the positive electrode. Smaller molecules move through the pores in the gel faster than larger molecules. This difference in the rate of migration separates the fragments on the basis of size. The nucleic acids in a gel matrix are invisible until they are stained with a compound that allows them to be seen, such as a dye. Distinct fragments of nucleic acids appear as bands at specific distances from the top of the gel, the negative electrode end, that are based on their size. A mixture of many fragments of varying sizes appear as a long smear, whereas uncut genomic DNA is usually too large to run through the gel and forms a single large band at the top of the gel. Figure 10.3. Shown are DNA fragments from 6 samples run on a gel, stained with a fluorescent dye and viewed under UV light. Polymerase chain reaction. DNA analysis often requires focusing on one or more specific regions of the genome. It also frequently involves situations in which only one or a few copies of a DNA molecule are available for further analysis. These amounts are insufficient for most procedures, such as gel electrophoresis. Polymerase chain reaction, or PCR, is a technique used to rapidly increase the number of copies of specific regions of DNA for further analysis. PCR uses a special form of DNA polymerase, the enzyme that replicates DNA, and other short nucleotide sequences called primers that base pair to a specific portion of the DNA being replicated. PCR is used for many purposes in laboratories. These include the identification of the owner of a DNA sample left at a crime scene, paternity analysis, the comparison of small amounts of ancient DNA with modern organisms, and determining the sequence of nucleotides in a specific region. Figure 10.4. Polymerase chain reaction, or PCR, is used to produce many copies of a specific sequence of DNA using a special form of DNA polymerase. Double-stranded DNA is denatured at 95 degrees Celsius. Primers are annealed at 50 degrees Celsius. DNA polymerase extends both DNA sequences. That occurs at about 72 degrees Celsius. And increased numbers of copies are made. And then the process continues. It cycles through. Cloning. In general, cloning means the creation of a perfect replica. Typically, the word is used to describe the creation of a genetically identical copy. In biology, the recreation of a whole organism is referred to as reproductive cloning. Long before attempts were made to clone an entire organism, researchers learned how to copy short stretches of DNA, a process that is referred to as molecular cloning. The technique offered methods to create new medicines and to overcome difficulties with existing ones. When Lydia Villa-Komaroff, working in the Gilbert lab at Harvard, published the first paper outlining the technique for producing synthetic insulin, diabetes researchers and patients received new hope in fighting the disease. Insulin at that time was only produced using pig and cow pancreases, and the life-saving substance was often in short supply. Synthetic insulin, once mass-produced, would solve that problem for many patients. These early discoveries led to the biotech boom and spurred continued research and funding for newer and better ways to improve health. Molecular cloning. Cloning allows for the creation of multiple copies of genes, expression of genes, and study of specific genes. To get the DNA fragment into a bacterial cell in a form that will be copied or expressed, the fragment is first inserted into a plasmid. A plasmid, also called a vector in this context, is a small circular DNA molecule that replicates independently of the chromosomal DNA in bacteria. In cloning, the plasmid molecules can be used to provide a vehicle in which to insert a desired DNA fragment. Modified plasmids are usually reintroduced into a bacterial host for replication. As the bacteria divide, they copy their own DNA, including the plasmids. The inserted DNA fragment is copied along with the rest of the bacterial DNA. In a bacterial cell, the fragment of DNA from the human genome, or another organism that is being studied, is referred to as foreign DNA to differentiate it from the DNA of the bacterium, the host DNA. Plasmids occur naturally in bacterial populations, such as E. coli, and have genes that can contribute favorable traits to the organism, such as antibiotic resistance, the ability to be unaffected by antibiotics. Plasmids have been highly engineered as vectors for molecular cloning and for the subsequent large-scale production of important molecules, such as insulin. A valuable characteristic of plasmid vectors is the ease with which a foreign DNA fragment can be introduced. These plasmid vectors contain many short DNA sequences that can be cut with different commonly available restriction enzymes. Restriction enzymes, also called restriction endonucleases, recognize specific DNA sequences and cut them in a predictable manner. They are naturally produced by bacteria as a defense mechanism against foreign DNA. Many restriction enzymes make staggered cuts in the two strands of DNA, such that the cut ends have a 2 to 4 nucleotide single-stranded overhang. The sequence that is recognized by the restriction enzyme is a 4 to 8 nucleotide sequence that is a palindrome. Like with a word palindrome, this means the sequence reads the same forward and backward. In most cases, the sequence reads the same forward on one strand and backward on the complementary strand. When a staggered cut is made in a sequence like this, the overhangs are complementary. Figure 10.5. A. In this 6 nucleotide restriction enzyme recognition site, notice that the sequence of 6 nucleotides reads the same in the 5' to 3' direction on one strand as it does on the 5' to 3' direction on the complementary strand. This is known as a palindrome. B. The restriction enzyme makes breaks in the DNA strand. And in C, the cut in the DNA results in sticky ends. Another piece of DNA cut on either end by the same restriction enzyme could attach to these sticky ends and be inserted into the gap made by this cut. Because these overhangs are capable of coming back together by hydrogen bonding with complementary overhangs on a piece of DNA cut with the same restriction enzyme, these are called sticky ends. The process of forming hydrogen bonds with complementary sequences on single strands to form double-stranded DNA is called annealing. Addition of an enzyme called DNA ligase, which takes part in DNA replication in cells, permanently joins the DNA fragments when the sticky ends come together. In this way, any DNA fragment can be spliced between the two ends of a plasmid DNA that has been cut with the same restriction enzyme. Figure 10.6. This diagram shows the steps involved in molecular cloning. Molecular cloning. Both foreign DNA and a plasmid are cut with the same restriction enzyme. The restriction site occurs only once in the plasmid in the middle of a gene for an enzyme, LAC-Z. The restriction enzyme leaves complementary sticky ends on the foreign DNA fragment and the plasmid. This allows the foreign DNA to be inserted into the plasmid when the sticky ends anneal. Adding DNA ligase reattaches the DNA backbones. These are recombinant plasmids. The restriction enzyme leaves complementary sticky ends on the foreign DNA fragment and the plasmid. This allows the foreign DNA to be inserted into the plasmid when the sticky ends anneal. Adding DNA ligase reattaches the DNA backbones. These are recombinant plasmids. The plasmids are combined with a culture of living bacteria. Many of the bacteria do not take any plasmids into their cells. Many take plasmids that do not have the foreign DNA in them, and a few take up the recombinant plasmid. The bacteria that take up the recombinant plasmid cannot make the enzyme from the gene that the fragment was inserted into, the LAC-Z. They also carry a gene for resistance to the antibiotic ampicillin, which was on the original plasmid. To find the bacteria with the recombinant plasmid, the bacteria are grown on a plate with the antibiotic ampicillin and a substance that changes color when exposed to the enzyme produced by the LAC-Z gene. The ampicillin will kill any bacteria that did not take up a plasmid. The color of the substance will not change when the gene for LAC-Z contains the foreign DNA insert. These are the bacteria with the recombinant plasmid that we want to grow. Plasmids with foreign DNA inserted into them are called recombinant DNA molecules because they contain new combinations of genetic material. Proteins that are produced from recombinant DNA molecules are called recombinant proteins. Not all recombinant plasmids are capable of expressing genes. Plasmids may also be engineered to express proteins only when stimulated by certain environmental factors, so that scientists can control the expression of the recombinant proteins. Reproductive cloning. Reproductive cloning is a method used to make a clone or an identical copy of an entire multicellular organism. Most multicellular organisms undergo reproduction by sexual means, which involves the contribution of DNA from two individuals, parents, making it impossible to generate an identical copy or a clone of either parent. Recent advances in biotechnology have made it possible to reproductively clone mammals in the laboratory. Natural sexual reproduction involves the union, during fertilization, of a sperm and an egg. Each of these gametes is haploid, meaning that they contain one set of chromosomes in their nuclei. The resulting cell, or zygote, is then diploid and contains two sets of chromosomes. This cell divides mitotically to produce a multicellular organism. However, the union of just any two cells cannot produce a viable zygote. There are components in the cytoplasm of the egg cell that are essential for the early development of the embryo during its first few cell divisions. Without these provisions, there would be no subsequent development. Therefore, to produce a new individual, both a diploid genetic complement and an egg cytoplasm are required. The approach to producing an artificially cloned individual is to take the egg cell of one individual and to remove the haploid nucleus. Then, a diploid nucleus from a body cell of a second individual, the donor, is put into the egg. The egg is then stimulated to divide so that development proceeds. This sounds simple, but in fact it takes many attempts before each of the steps is completed successfully. The first cloned agricultural animal was Dolly, a sheep who was born in 1996. The success rate of reproductive cloning at that time was very low. Dolly lived for 6 years and died of a lung tumor. There was speculation that because the cell DNA that gave rise to Dolly came from an older individual, the age of the DNA may have affected her life expectancy. Since Dolly, several species of animals, such as horses, bulls, and goats, have been successfully cloned. There have been attempts at producing cloned human embryos as sources of embryonic stem cells. In the procedure, the DNA from an adult human is introduced into a human egg cell, which is then stimulated to divide. The technology is similar to the technology that was used to produce Dolly, but the embryo is never implanted into a surrogate carrier. The cells produced are called embryonic stem cells because they have the capacity to develop into many different kinds of cells, such as muscle or nerve cells. The stem cells could be used to research and ultimately provide therapeutic applications, such as replacing damaged tissues. The benefit of cloning in this instance is that the cells used to regenerate new tissues would be a perfect match to the donor of the original DNA. For example, a leukemia patient would not require a sibling with a tissue match for a bone marrow transplant. Frieda Miller and Elaine Fuchs, working independently, discovered stem cells in different layers of the skin. These cells help the skin repair itself, and their discovery may have applications in treatments of skin disease and potentially other conditions, such as nerve damage. Figure 10.7. Dolly the sheep was the first agricultural animal to be cloned. To create Dolly, the nucleus was removed from a donor egg cell. The enucleated egg was placed next to the other cell. Then they were shocked to fuse. They were shocked again to start division. The cells were allowed to divide for several days until an early embryonic stage was reached, before being implanted in a surrogate mother. Genetic engineering. Using recombinant DNA technology to modify an organism's DNA to achieve desirable traits is called genetic engineering. Addition of foreign DNA in the form of recombinant DNA vectors that are generated by molecular cloning is the most common method of genetic engineering. An organism that receives the recombinant DNA is called a genetically modified organism, or GMO. If the foreign DNA that is introduced comes from a different species, the host organism is called transgenic. Bacteria, plants, and animals have been genetically modified since the early 1970s for academic, medical, agricultural, and industrial purposes. These applications will be examined in more detail in the next module. Although the classic methods of studying the function of genes began with a given phenotype and determined the genetic basis of that phenotype, modern techniques allow researchers to start at the DNA sequence level and ask, what does this gene or DNA element do? This technique, called reverse genetics, has resulted in reversing the classical genetic methodology. One example of this method is analogous to damaging a body part to determine its function. An insect that loses a wing cannot fly, which means that the wing's function is flight. The classic genetic method compares insects that cannot fly with insects that can fly and observes that the non-flying insects have lost wings. Similarly, in reverse genetics approach, mutating or deleting genes provides researchers with clues about gene function. Alternately, reverse genetics can be used to cause a gene to overexpress itself to determine what phenotypic effects may occur.